Hello dear learners, welcome to my channel Microbiology Tutorials. I am your instructor Anshul Kumar, and today I am going to present on the topic immunology and oncogenesis from elimination to escape pathway. Most of the medical students would be knowing that the role of immune system is to eliminate the foreign pathogens and foreign antigens from the host cells to remain the entire body and host system healthy and free from any infections or dangerous pathogens. Now, mostly we know that how the immune responses in the cases of foreign substances which enters or evades our host cells. In this chapter, we will discuss that how the immune system reacts when, in the case of oncogenesis or malignancy conditions develops. And we know that these malignant conditions can either maybe due to some tumor causing viruses or bacteria or other foreign agents like some radiations, chemical exposure to some chemicals, then drugs, or it may be due to mutations in the cells which removes the control mechanism. Usually, when we discuss about the oncogenesis, it means there is an overgrowth of cells and tissues in the body, and it basically occurs due to unchecked or uncontrolled growth in the body. So we will see that does our immune response or immune system checks any type of irregular growth or any abnormal growth in the body, or does it is or does these malignant cells escape this immune mechanism, or immune system fails to recognize it. So it is basically, if I go through the keywords, I can classify this in some basic terms associated with it. So first is the most important is the immunosurveillance. Immunosurveillance means that our immune system holds check on any type of abnormal growth in the body, and it checks any type of disorders associated with it. Next is the immunotolerance. Immunotolerance is usually when these malignant cells escape the route of immunosurveillance, and the immune system recognizes it as self-antigens or self-cells and do not act against it. And it, in conjunction with the normal cells, it also maintains in equal growth. In some cases, even immune cells, along with some other components of the body, they create a tumor microenvironment which helps in the progression of these tumor cells. Means It supports the growth of these tumor cells. And and all the most of the innate and adaptive immune cells are involved in this tumor progression. And finally, the it advances, the cancerous conditions gets into an advanced stage because immune system stops acting against it. The third important term is the process of immune editing. We will see what is the immune editing in our chapter, in our lecture here, and how from eliminating to accepting it as its own. In this process, the immune cells react or interact with the tumorigenic cells. Next, the important term are the most important is the tumor associated antigens and tumor specific antigens. Here, tumor associated, if I differentiate tumor associated antigens and tumor specific antigens, as the name itself suggests, that here tumor associated antigens are present more on both, mostly on the tumor cells, but they are also present on normal cells. But the only difference is that in normals, in tumor cells, they are overexpressed, whereas in the normal cells, they are not expressed so widely, but they remain in check. Whereas tumor-specific antigens are mostly associated with the tumor-specific cells. So here, basically, we see that tumor cells which develop immune tolerance are mostly associated with tumor-associated antigens because it is easier for the immune cells to recognize it as self cells in comparison to the tumor specific antigens, which are highly immunogenic. Here, immunogenic means that immune response are triggered more in case of tumor specific antigens, and chances of eliminating it is more than in tumor associated antigens. So with this some in a brief, I gave you the overall summary of this chapter, and in detail we will see through this while I will continue through the different parts of this lecture. So coming to the immunology in oncogenesis, it is a complex interplay where the immune system attempts to eliminate tumor cells immunosurveillance, while tumor cells evolve to evade detection, that is immune evasion. So it is basically a hide and seek like play where immune cells try to destroy. It is in all the cases, in all any immune system immunity we see, it is basically the same. The foreign particle or any abnormal part of in our body will try to escape the immune system of the body, whereas immune system will try to recognize it and destroy it. So when the elimination takes place, that the term is used as the immunosurveillance. While tumor cells try to evolve to evade detection, that is the immune evasion, and this process, the entire process is known as immunoediting, which operates in three phases: elimination, equilibrium, and escape. Three E's we can say here. We will see what does it mean, and ultimately shaping the tumor microenvironment means that it will develop an ecosystem which facilitate the progression of the tumor cells, which is also called as tumor in microenvironment, and very different type of cells. Even the angiogenesis, new vascular capillary blood vessels also develop for them um, to develop or migrate to other parts of the body from the site of development, and hence all this will create a proper environment for the cancerous cells to progress and influence cancer progression or regression. Key immunology aspects in oncology is immunoediting in three phases, that is the elimination, equilibrium, and escape. And persistent interaction leaves only less immunogenic cancer cells, which then enter the escape phase, establishing a tumor that evades surveillance. Now here through the diagrammatic representation, you can understand here that the yellow dots which are the normal cells. So here, if we see here that environmental factors and due to genetic mutations, they are converted. Some of this cells has been convert transformed, or we can say that they have been transformed into tumor cells, and hence the immune system of the body develops to eliminate these transformed cells. And here again, here we can see that blue dots, weak immunogenic and immunoevasion transformed cells, and it gets transformed into weakly immunogenic cells, and it creates a equilibrium state with the normal cells. And after that, suddenly, if the immune system stops or ceases to react against them, and they escape this entire immune response, either it may be an innate and both adaptive immunity, and hence carcinogenesis or oncogenesis progresses. So this through this diagram you can easily understand that how the process of immunoediting takes place. Now next here we can say that anti-tumor immunity, how it acts. So basically the most player here, or the most important cells which are involved are, first is the cytotoxic T lymphocytes, CTLs in short, or CD8 plus cells, which are the principal mediators which recognize tumor antigens on MHC1, kill via perforin and granzymes and FAS and FASL. Then in the innate immunity, we can see natural killer cells, then macrophages. Here macrophages, here you can see that M1 type cells. See macrophages, if we say that they are divided into M1 type and M2 type, if strictly if we see the oncogenesis, M2 type cells are mostly creating the tumor microenvironment. Means they are anti-inflammatory. Means slowly it helps in the progression of the tumor cells. In normal conditions, they act as a cell which helps into heal any wound or infections. Whereas macrophages M1 type are the pro-inflammatory cells, means the cells which participate in phagocytosis, release tumor necrosis factor alpha, nitric oxide reactive oxygen species activated by interferon gamma. Then next cells is the helper T cells, then antibodies, which helps in the opsonization, complements activation, and antibody dependent cell mediated cytotoxicity. And these are the some of the key cytokines: interferon gamma, TNF alpha, IL-2, and IL-12. Now the immune response to cancer cells also be separated into innate and adaptive immunity. And in adaptive immunity, both humoral and cellular components are involved. Innate immunity in general are non-specific response to pathogens or cancer cells mediated by antibodies and cellular elements, including dendritic cells, macrophages, natural killer cells, and natural killer T cells. Thereafter, adaptive immune mechanism largely mediated by cytotoxic T lymphocytes facilitate recognition of distinct tumor antigens as foreign and destruction of tumor cells. Most humoral responses cannot prevent tumor growth. Usually, we see that the most important cells which are involved in suppression of these tumor cells are the cytotoxic T lymphocytes, which are the part of cell-mediated immunity. Whereas we all know that humoral responses are constituted by antibodies. So antibodies have minimal role, though they participate in this immune mechanism, but the majority of the functions are performed by the cytotoxic T lymphocytes. However, effector cells such as T cells, macrophages, and natural killer cells have relatively effective tumoricidal abilities. Now, effector cell activity is induced by other cells that present tumor-specific antigens or tumor-associated antigens on their surface. And these cells are called antigen-presenting cells and is supported by cytokines. Despite the activity of effector cells, host immunoreactivity may fail to control tumor occurrence and growth. Coming to the cellular immunity, the T cell is the primary cell responsible for direct recognition and killing of tumor cells. As I already discussed, T cells carry out immunological surveillance, then proliferate and destroy newly transformed tumor cells after recognizing tumor-associated antigens. The T cell response of tumors is modulated by other cells of the immune system. Cells require the presence of humoral antibodies directed against the tumor cells to initiate the interactions that lead to the death of tumor cells. In contrast, suppressor T cells inhibit the immune response against tumors. Cytotoxic T lymphocytes recognize antigens on target cells and lyse these cells. These antigens may be cell surface proteins or maybe intracellular proteins that are expressed on the surface of combination with class 1 major histocompatibility complex. Here, one thing that MHC here for cytotoxic T lymphocytes class, it is necessary that for function of T cells, it is very necessary that these antigens should be presented by antigen presenting cells and that is expressed on the surface in combination with MHC or a major histocompatibility complex, which is for cytotoxic T cells, it is class 1, and for helper T cells, it is class 2. Now here you can see some of the important examples of tumor-specific CTA cells which are found here in some of the cancer conditions, colon, endometrial, kidney cancers, and all those which have been written here. Then natural killer cells after cytotoxic T lymphocytes. Another important cell is the natural killer cells, which are a subset of T cells only, but they are non-specific, not as a cytotoxic T lymphocytes, and they do not require presentation of antigens, but they react non-specifically. Effector T cells with anti-cancer activity, in contrast to CTLs, NK cells lack antigen receptors but can recognize normal cells infected with viruses and cancer cells. Their tumoricidal activity is termed natural because it is not induced by a specific antigen. The mechanism by which NK cells discriminate between normal and abnormal cells is under study. It is still to be evaluated how they recognize or how they able to differentiate between normal and abnormal cells. Evidence suggests that class 1 MHC molecules on the surface of normal cells inhibit NK cells and prevent lysis. Thus, a decreased level of class 1 MHC molecule expression characteristics of many tumor cells may allow activation of natural killer cells and subsequent tumor lysis. Another are natural killer T cells are specialized CD1D restricted T cells that recognize lipid antigens. CD1D is a member of the CD1 group of glycoproteins on the surface of human antigen-presenting cells. Following stimulation, natural killer T cells lead to activation of both innate and adaptive immune cells in the tumor microenvironment. Natural killer T cells are innate like T cells that quickly respond to antigenic stimulation and rapidly produce large amounts of cytokines and chemokines. Then macrophages, next important cells which are involved in the tumor oncogenesis are the macrophages, which kill specific tumor cells when activated by combination of factors, including lymphokines. Lymphokines are the soluble factors which are produced by T cells and interferons. They are less effective than T cell mediated cytotoxic mechanism under certain circumstances. Macrophages may present tumor associated antigens to T cells and stimulate tumor-specific immune response. There are two polarized states of tumor-associated macrophages, TAM1 and TAM2, as I already discussed that what is the role of M1 cells and M2 cells. So M1 and M2 are considered to exist on a continuum until they maximally differentiate into M1 and M2. Polarization can vary over time and depend on their location inside the tumor and the stage and the type of cancer and prior treatment. Now, next cell, that is the dendritic cells, are dedicated antigen presenting cells present in barrier tissues. They are all the first line of defense. All these macrophages, dendritic cells, natural killer cells, they are part of the innate immune system and acts as a first line of defense mechanism. So skin, lymph nodes, they play a central role in initiation of the tumor-specific immune responses. These cells take up tumor-associated proteins, process them, and present to T cells. So they act as antigen-presenting cells, which helps in the stimulation of CTL response against tumor. Lymphokines, already we have studied that lymphokines are the soluble factors produced by T cells, stimulate growth or induce activities of other immune cells. Such lymphokines include the interleukin 2, also known as T cell growth factor, and the interferons, interleukin 12, produced by dendritic cells, and specifically induces CTLs, thereby enhancing anti-tumor immune response. Regulatory T cells are normally present in the body and help prevent autoimmune reactions. They are produced during the active phase of immune response to pathogens and limit a strong immune response that could damage the host. Accumulation of these cells in cancer inhibits anti-tumor immune responses. So regulatory T cells helps in the inhibition of anti-tumor immune response and development of the tumor cells in the host cells. So then is the myeloid-derived suppressor cells, then granulocytic, monocytic, two populations of these cells are there, and myeloid-derived suppressor cells accumulate in large numbers in cancers and predict poor clinical outcomes in various types of cancers. Next is the humoral immunity, basically constituted by the antibodies. So in contrast to T cell cytotoxic immunity, humoral antibodies do not appear to confer significant protection against cancer. Most antibodies cannot recognize TAs. Regardless, humoral antibodies that react with tumor cells in vitro have been detected in the sera of patients with various tumors. So they are important in the diagnostic part, as we can identify the important in the serum, the antibodies against those tumorogenic antigens. So they play an important role in diagnosis of this tumor oncogenesis. So these are some of the important antibodies found in the sera: breast cancer, gastrointestinal tract cancer, lymphomas, lung cancers, melanoma, and others as we have mentioned here. Next, the cytotoxic antibodies are directed against surface antigens of tumor cells. These antibodies can exert anti-tumor effects through complement fixation or by serving as a flag for destruction of tumor cells by T cells. Antibody-dependent cell-mediated cytotoxicity, ADCC. So both either through complement system or through activation of T cells, another by these antibodies. Another population of humoral antibodies called enhancing antibodies or the blocking antibodies may actually favor rather than inhibit tumor growth, as we have seen in the regulatory T cells here, which support the TME, that is the tumor microenvironment, is the enhancing antibodies. The mechanism and relative importance of such immunological enhancements are not well understood. Antibodies that deliver conjugated toxins to tumor cells to selectively target tumor cells are being tested. Next, how there is a failure of host defenses. Although some tumors are eliminated by the immune system, others continue to grow despite immunosurveillance. Several mechanisms are proposed. What are they? They are a specific immunological tolerance to TAs in process that involves antigen presenting cells and suppressor T cells, possibly secondary to prenatal exposure to the antigens. So there is development of tolerance against these tumorigenic antigens in the body, and usually it possibly secondary to prenatal exposure. That is the before birth it takes place. Suppression of immune response by chemical, physical, or viral agents. Helper T cell destruction by human immunodeficiency. Virus, one of the most important, uh, the retrovirus, which helps in the development of this oncogenic conditions. Then suppression of immune response by cytotoxic drugs or radiations. So all these suppression systems through the different modes, either through exposure to radiations, chemicals, viruses. So usually we see that the immune system is lowered, or their functions decreases, and it gives an opportunity for such tumor cells which are not in large numbers to overgrow, and as such they develop those the oncogenic conditions or malignant conditions in the body in with the support of these pathological agents, as well as these different environmental factors which are also responsible. Now here, this suppression causes various problems, including decreased T cells, B cells, and antigen presenting cell functions. There is a failure of the immune response to different type of infections, and presence and activity of tumor-associated macrophage 2 polarized cells, myeloid-derived suppressor cells, and regulatory T cells. So all these cells create a helpful environment for the progression of the oncogenic cells. Now, according to immune surveillance theory, the immune system patrols the body to recognize and destroy cancer cells. However, the occurrence of malignancies indicate that this protection is not perfect. Moreover, the many mechanisms exhibited to counteract immune response imply that malignant tumors undergo a selection process established by the immune system. Accordingly, a modified theory, cancer immunoediting theory, has been proposed, taking into account the immune system not only can protect the host against tumors, but also by selecting the tumors of lower immunogenicity has the capacity to shape and even promote the generation of new variants, displaying increased ability to escape and overcome immune control. Now, immune response to cancers. So, immune response to cancer is best viewed as specialized case of immunity in which the malignant cells have adapted and learn how to persist. The immunological surveillance theory was originally put forth independent by Burnett and Thomas, who suggest that immune system continually surveyed the body for the presence of malignant cells, which were continuously arising as a result of mutations. So, according to Burnett and Thomas, the primarily the responsibility of development of these tumor cells or the malignant cells was due to arising as a result of mutation. So mostly mutation is responsible for these cells, malignant cells. And potential strength of immune surveillance is based on the knowledge that cancer cells do not indeed express TAs or TSAs that can be recognized by the immune system as foreign elements. So antigens also play a major role in recognition of these tumor cells. If they are not recognized, or they are recognized also but as self antigens, then immunosurveillance will not take place, and these tumor cells will develop immunotolerance in between them. Then development of cancer cells can be explained by the ability of tumor cells to evade immune recognition either by the failure of immune system or to be adequately generated or by the induction of immune tolerance or other inhibitory mechanism that allows tumor to escape immune detection and elimination. Now, in contrast, the tumors induced by carcinogens, in which each new tumor has unique antigenic specificity regardless of their morphologic appearance, tumors induced by viruses constantly consistently express antigens that cross-react with other tumors induced by the same or similar viruses, even though their morphologic appearance may differ. So this is the basic difference between the tumors caused by any carcinogenic agent or and on the other place the tumors which is induced by the viruses. Now, cancer immunoditing is a multifaceted process that encompasses the well-known threes, namely elimination, equilibrium, and escape. As we have already discussed, you can see here through the diagram that how here innate immune response and adaptive immune response in the phase of elimination, they act very actively and try to eliminate, which can be demonstrated here through the pink cells and green cells. In the first stage, the pink cells are predominant, whereas green cells are very less. Whereas in the phase of equilibrium, you can see that suddenly there is a fall in the immune response against these malignant cells or tumor cells, and it coexists along with the normal cells. Here you can see here that both pink cells and green cells are equally present in the body. And suddenly in the escape phase, you can see here that there is a they escape this immunosurveillance mechanism, and their counts suddenly increases and they overpower the normal cells. And this is the phase where there is a advancing of the tumor cells. So elimination, equilibrium, and escape are the three phases of the immunoediting process in the malignant conditions. So here I have already described you through this diagram. Here you can see here how there is a intrinsic and cell intrinsic immune system. In the first phase, you can see here that if I see here through the pyramidal structure, in the immunosurveillance stage, the immune system is acting very predominantly and keeping the tumor cells in check. Suddenly, in equilibrium phase, there is an immunoselection phase where both the normal cells and malignant cells occur, and this is the advanced oncogenesis stage. And in the escape stage, suddenly the immune response subsides and it leads to the proliferation of the tumor growth. So this is also a very well explained diagrammatic representation of immune response against the tumor cells. Now, cancer immunosurveillance versus immunoediting. The basic difference is that the cancer immunoediting process is envisaged to consist of three phases: elimination, equilibrium, and escape, where elimination phase corresponds to the original concept of cancer immunosurveillance, whereby nascent tumor cells are successfully recognized and eliminated by the immune system, thus returning the tissues to their normal state of functions. Now, tumor cells that elude the immunosurveillance phase will progress to the immune editing phase called the equilibrium phase of advanced oncogenesis, where tumor expansion and metastasis are minimal and usually occur without symptoms. In the equilibrium phase, that immune system may eventually eliminate all tumor cells, leading to an outcome similar to the elimination phase. In a second scenario, the constant interaction of the immune system with tumors over a long period of time may actually edit or sculpt the phenotype of the developing tumor, resulting in the immunoselection of a tumor that has been shaped into a less immunogenic state. Tumors that are no longer susceptible to immune attack then progresses to the immunoediting process termed as escape phase. And the emergence of clinical symptoms of cancer generally correlates with the escape stage. Tumor subverts the immune system either directly through its non-immunogenic phenotype or indirectly through a variety of immunosuppressive mechanism. And through this diagrammatic representation, you can see the different stages of immunoediting, how it takes place, the cancer immunoediting. And this is the cancer immunity cycle, whereas an anti-cancer immune response involves a cycle starting with a release of cancer antigens captured by dendritic cells, priming T cells, and finally killing cancer cells, typically by cytotoxic T lymphocytes, an immune evasion mechanism, where the tumors evade immunity by down-regulating MHC1 molecules to hide from T cells, upregulating checkpoint molecules to inhibit T cell activity and creating an immunosuppressive microenvironment. So we can say in briefly, we can say that if the till it is recognized as foreign or foreign pathogen or we can say that abnormal cells, immune system work efficiently to eliminate all these tumorigenic cells. But once it suddenly it subsides the immune system or skips this recognition or immunosurveillance mechanism, then it starts progressing and also involves different type of immune cells such as regulatory T cells or TAM2 and other cells which helps in this their progression instead of eliminating them. And different type of viruses, basically oncogenic viruses or radiation exposure or mutations, these all contribute in the subside sub or decrease activity of the immune cells. And in addition to the in this type of infections, the chances of developing malignancies also increases. So I hope this lecture would have been beneficial to all of you, and you would have learned substantially from my lecture. If you like this lecture, please do like and subscribe so that I keep myself motivated and bring some more information for you. With this, I conclude my today's lecture. Thank you, all of you.